this. So I got a bunch of emails from folks about the likelihood ratio test. Sorry if it was later last night, I couldn't respond. I was driving. So anyway, so just to give you a heads up about that, right? So the idea there is that, here, let me write this out so you guys will probably understand it a little better. So it's again one of those things, if you feel like you weren't able to finish up the likelihood ratio test, just go ahead and submit it again later. That's fine. Excuse me. Happy birthday. You said? Oh, well, happy birthday. So the idea behind the likelihood ratio test is actually harkens back to Bayes' theorem again. So let's assume that you have here, you guys see here. Let's assume that hypothesis 0 is your null, let's say, or let's just call it as one hypothesis, and then HA is your alternate hypothesis. And D is your data, right? So whatever your data is. So the probability of your hypothesis 0 given your data, right? This is called the likelihood, right? So this is the likelihood in this case of hypothesis 0. And you can compute this, right? So let's say that you constructed a tree using mega, and it will give you a log likelihood value. That is this one, okay? And let's say that in this case, this might be, I don't know, GTR, all right? So you use the GTR model, and let's say your null hypothesis is that it is the GTR model. And let's say the alternate hypothesis happens to be the Jukes-Cantor 69 model, okay? That was the substitution model that you were using. So then similarly, you would get a probability of the alternate hypothesis, right? Some value right here, right? So the thing about the likelihood ratio test, by the way, these are already log likelihoods, right? So you don't have to compute the log on top of that. Okay. Well, actually, if you try to compute it, it will crash. It won't work, right? Because you can't get a log of a negative value anyway. So, okay. If you compute -2 times the log likelihood ratio. So in other words, -2 times log of the probability of H0 given D divided by log of the probability of HA given D, right? This, as it turns out, is the expectation of this distribution happens to be chi-squared distributed, okay? So which means that computing this value, what you can try to do is you can try to fit this to the chi-square distribution with a certain number of degrees of freedom. So the degrees of freedom will be computed as just the total number of additional parameters or total number of additional degrees of freedom added by the alternate hypothesis compared to the null hypothesis, or vice versa, okay? So in this case, right, let's say, I can't remember how many degrees of freedom I said the GTR has, 10 or something like that. Yeah. And Jukes-Cantor model, I think, has 2, maybe. I can't remember. Was it just 1? Yeah, I think it was just 1, right? Because it's equitable probability of everything. So in other words, if you're testing GTR versus Jukes-Cantor, the total number of degrees of freedom would be 10 - 1, which is 9, right? Because you have, that is the total difference in the number of parameters between one model versus the other. And that gives you the total number of degrees of freedom. So then if you're testing this versus that, right? In this case, example, right? This would just be 10 - 1, which is equal to 9. So then you can easily compute this value, and you know this value. So then you can go up and look up a chi-square table to obtain the critical value from the chi-square distribution at a certain, say, p-value cutoff. Let's say I'm going to use a p-value cutoff of 0.05, right? That's normal sort of rule of thumb. Everybody uses that. So let's see what that tells you, right? So let me stop sharing here. Let me pull up a chi-square table, let's say. So, sorry. I just Googled chi-square table. I'm sure you can find one easily. So here's one, right, that gives me all the critical values of the chi-square distribution and various degrees of freedom, right? So whatever chi-squared value that you compute from your likelihood ratio, and then what you would do is, let's say in this case, I have 9 degrees of freedom. So you would just look up across the p-value distribution up here. And let's say I'm going to use a 0.05 cutoff. In this case, if let's say your value happens to be 20 point something, right? In that case, 20 point something is actually greater than this value, right? Which means the p-value would be less than 0.05, right? So then you would say that there seems to be greater support in this case for the Olsen hypothesis compared to the null hypothesis. If it's less than 0.05, then you would say that there's greater support, or there is no additional support for the Olsen hypothesis compared to the null hypothesis. That's all. Okay, does that make sense? So that's what you will do when you do the likelihood ratio test. And Mega prints that out, right? Mega gives you the likelihood value for that particular tree. So you would use that to manually compute the likelihood ratio. Cool. If you were not able to finish that up as part of the assignment, just go ahead and do it and submit it. That's fine. I'm probably not going to get to grading them until later this week anyway. I did finish grading up your exams, though. Good job. Everybody did great. I can't even remember what the average was, like 90 or something. You're all rocking this. So Will, you had a question. Go ahead.
Yeah. So for the likelihood ratio test, which 2 data sets are you comparing?
So in this case, you are comparing, it's the same data set, right? You build, so let's say you built it based on the protein sequence, the amino acid sequence, right? And you built a likelihood tree based on, let's say, Jukes-Cantor model. And then you built the same likelihood tree based on, let's say, the GTR model. So it's the same data set,
Oh,
two different models.
Got you.
Does that make sense?
Yeah.
Yeah, you can't compare 2 different data sets, right? Because that would be apples to oranges. It's the same.
Okay.
It's the same data using different models. Okay. And that's the only scenario when the likelihood ratio will be chi-square or expected to be chi-square distributed, because you can't expect 2 separate data sets and then compute the likelihood ratio between that. You can't do that. In that case, we do something else, right? There's other kinds of information criteria that can be used to pick models. If you're actually using just completely wildly different, data sets and models and whatnot, right? So that's a completely different way of selecting models. Did you have a question? Sorry, Kia.
Oh yeah. So in your example, it goes from the ratio to 2 times like L and the model 1 minus model 2.
Mm-hmm.
What happens to the negatives? Are we not?
So it's already calculated, right? So you
So you don't have to take that out.
Yeah, so Mega already prints out
Yeah.
the likelihood ratio, which would be negative anyway.
Oh, so they all they cancel out.
Correct.
Okay,
It'll just cancel out. Okay. But remember, you have to calculate minus 2 LLR, right? So it's minus 2 multiplied by that value.
Yeah.
Whatever that is. Okay. Yeah. Okay. Oh, actually, you know what? It would be, because it's already calculated as negative, I think. So it would be just 2 LLR, right? Let me double check, actually. That's a good question. This was where I picked up the example. Oh, it's just 2 LLR. It's not minus 2. Sorry.
Oh, okay.
Great. Any other questions about the assignment? Hopefully that was straightforward. Mega is very easy to, very intuitive. It's a pretty nifty. Here's one example of tree that I had built. In this case, in my case, it predicted gorilla with Homo sapiens, and then Peniscus after that, and then I had the macaque after that, I think. But whatever sequence that you used, the tree might vary, and what model you used might also vary the tree. Yeah. No. Yeah. Sometimes they will not match, right? It all depends on how Mega computes its neighbor joining, algorithm, whatever algorithm it uses. The one algorithm we discussed, right, the Fitch-Margoliouth, or actually the one we discussed is sort of like a simplified version of the Fitch-Margoliouth algorithm. I don't know what neighbor joining algorithm Mega uses. So for all you know, the trees might be different. So don't stress. The UPGMA tree for the most part should be the same, I think, if I remember right. At least the topology of the UPGMA tree should be the same. Because the UPGMA tree is just, it's just literally just computed based on just taking the distances and dividing it by 2, right? So it's very easy to compute those distances. So I think those should be the same. The neighbor joining might be different, And likelihood might be wildly different. Again. Sound good? There aren't too many differences between these 4 or 5 sequences, by the way. They are very highly similar to each other. Right? I mean, these are great apes anyway, right? So we're all relatively recently related by descent, right? 1 million years or 2 million years at the most, right? So it's not too crazy. So they should be very similar to each other anyway. Great. Sorry, I didn't realize I wasn't sharing my screen here. I was going to show you guys my tree that I have. Sorry. Here, this was the tree that I had. So I had Homo sapiens with gorilla, and then Piniscus after that, and then macaque after that. So, but again, I don't remember what sequence I used. I don't know if I used nucleotide or amino acid sequence. You can use either, by the way, right? But the only difference is that the mutation model would be different, right? Compared to if you're using protein sequences, amino acid sequences versus DNA sequences. Cool. By the way, I also, my 2000 sequence, whatever, Raxamel run also finished up almost right after my class finished up. I went back to my office and it was done. And then I pulled it up using Victory and it looks kind of ugly. So I'm gonna try and clean it up a little bit so I can actually show you guys what this looks like. But this was my tree. Obviously there was like thousands of sequences, right? So it looks really ugly. But I'm gonna try and clean this up a little bit so I can actually show you guys what these sequences essentially look like. But your tree run should have also finished, I think, over the weekend at least. By the way, I'm using Victory here. There's a link to Victory on your lab manual's last page. So you can download. Victory is a really nice software where you can actually make edits to the tree, how to visualize it. You can, you have options for sort of either making it sort of like, this kind of a rectangular thing, or maybe you can make it a radial tree, et cetera, et cetera. It's a pretty cool piece of software. So do check it out, okay? Okay, so, all right, so we've discussed a lot of things so far, right? So we've discussed how we start out with sort of next generation sequencing data, right? We start with reads, we assemble those reads, we've talked about assembly algorithms. And then after that, we talked about sort of lining them up against each other. We've also talked about lining them up against the reference genome, how to compute sort of the Burrow-Wheeler transform and suffix array based algorithms for discovering the location of a particular read. Based on those values, we've also talked about variant calling algorithms, right? So we talked about how to compute genotype likelihoods or variant calling likelihoods. Then we talked about phylogenetic algorithms, and there we talked about genetic distance-based methods, parsimony methods, and likelihood-based methods, right? So, so the next sort of obvious next step, right, is how to do population genomic analyses based on these data sets, okay? So we're going to discuss, spend quite a little bit of time, again, by virtue of the fact that I'm a population genomicist, kind of stuck with me, but that's okay. Because population genomics is sort of the idea that having observed similarities and differences, how do you make sense of it, right? What kind of evolutionary processes are actually occurring as these sequences are evolving over time and space, right? So that's kind of the big picture idea of population genetics and population genomics. So we're going to learn various population genomic methods that can be applied to these either multiple sequence alignment data sets or variant data sets and make inferences based on that, okay? Now, some of these concepts might be familiar to a lot of you, but if not, great, we'll just review all of these concepts nonetheless, okay? So that's what we're gonna spend a little bit of time kind of doing. Some of this is pretty heavy on the theory, so please bear with me at any point if you're stuck or if you have questions, please stop me and ask. Because if you don't ask questions, I'm just gonna assume that you're good with it and I'm just gonna proceed, okay? Because some of it might involve some calculus, some of it, some algebra, stuff that hopefully folks are familiar with regardless, but at any point, let me know and we'll stop, okay? Okay, so first things first, let's go ahead and review some concepts, okay? So let me share my iPad screen here again. Let me pull up my notes here so I can go over the different terms for my own sanity. Okay, so let's talk about some concepts first. Okay, so let's start with some nomenclature. Okay, we're going to start with. So by the way, this is just population genomics. So let's start with some nomenclature. The first one we're going to use is a locus. So again, we might have discussed this earlier on, but tell me, what is a locus? Anybody?
Particular region on the chromosome.
Yeah, it's some physical location on a chromosome, right? So physical location on a chromosome is called a locus, right? Now, the most common sort of misconception that I hear about is folks confuse locus with a gene, right? So they are not the same, right? Because a gene is much more specific, because a gene essentially refers to a protein coding locus. That's all. Okay, that's the only difference really if you think about between a gene and a locus, right? So a gene would be a protein coding locus. Okay, so that would be a gene. So then, right, the next obvious question then is, what are the different variants at a locus? And we call these as alleles, right? So an allele is essentially a variant at a locus. And when I say variant, this could be of different types. We've already discussed different types of variants or mutations. By the way, mutation, variant, allele, polymorphism, they're all the same, right? So they all refer to the same concept or idea, right? Because you wouldn't have variants or alleles if you didn't have mutation, right? So these could be single nucleotide polymorphisms like A, C, G, Ts. They could be indels, right? Insertions and deletions. It could be microsatellite or mini satellite markers. They're basically tandem repeat regions. So you could just use the counts of the number of those repeats as an allele, right? Or they could also refer to things like transposable element, copy number variants, et cetera, right? So all of those encompass this idea of being an allele. So any variant at a locus is what is referred to as an allele. So then if you take the allelic combination at a locus, we call that as a genotype, right? So the combination of alleles, so I'm just going to call it combination of alleles at a locus in an individual. We would refer to this as a genotype, right? So for instance, right, if you say that at any one particular location in a diploid individual, this individual happens to be AA, right? That means both copies, maternal paternal copies, happen to be A at that locus. We would call that as an AA genotype. Now, on the other hand, when we think about, say, things like viruses, right? So SARS-CoV-2 genome, for instance, happens to be haploid, right? Which means there's only one copy of an allele at that particular locus. We would call that as a haplotype. So the allele at that locus and all of the alleles at neighboring loci together, as if you take it as a chunk of that genome, we would call that as a haplotype. That's all. Okay. So haplotype is just, we would call this combination of alleles at neighboring loci, right? So, and this is very loosely sort of defined. Right? It could be, 10 bases, it could be 10 kilobases, whatever it is, right? So it's just a chunk of loci at a time. You take all the allelic specifications at that, and this has to be haploid, right? So it's just one single locus. Speaking of, right, Ploidy is another term. So what is ploidy? Correct, right? So number of homologous chromosomes or number of sets of homologous chromosomes in a single cell, right? So we call this as number of sets of homologous chromosomes in a cell, right? So we call that as ploidy, okay? So haploid meaning one single set, diploid meaning, or diploid, I don't know how people call it, I say diploid. Diploid meaning two sets of chromosomes, triploid, quadruploid, pentaploid, or anything beyond that we just call polyploid, right? So polyploidy is very common, for instance, in plants, right? So polyploid means multiple sets of chromosomes. Great. Now let's talk about some. Let's talk about say one more concept. Let's add linkage to this. What is linkage? Yeah, right. So our loci. Let's make it much more simpler, right? So it's not just genes, right? It's loci, right? So when you have loci and allelic combinations of those loci inherited together, and why are they inherited together? Because they're close together, right? On the same chromosome, right? So alleles at multiple loci on the same chromosome, right? That's important. That are inherited together, right? We call that as linkage. When are they not inherited together? When you have recombination, right? So when you have homologous recombination, right? So homologous recombination refers to sort of this breakage of this linkage, right? So when you have recombination, you have segments of homologous chromosomes exchange between homologous chromosomes during meiosis. And so that, when that happens, alleles are inherited separately or independently. So this is just exchange of homologous segments during meiosis, right? You guys remember which phase of meiosis recombination occurs in? Yeah, prophase 1, that's correct, right? So it occurs during prophase 1 of meiosis, specifically during prophase 1. Great. So then this leads to sort of some simple Mendelian principles, again, harkening back to your genetics classes, right? That there's 3 Mendelian principles of inheritance, right? And these are, again, we call these as Mendelian principles because they only hold true for Mendelian type of loci or Mendelian types of genes, okay? So there are 3 Mendelian principles. So let's talk briefly about Mendelian principles. This is very important in population genetics. So the first principle is what we call the principle of dominance. And the principle of dominance states that there seems to be, sort of some simple dominance relationship between alleles at a single locus, right? So one allele seems to show dominance, which means that it seems to be present at a greater number in that population compared to others. Okay, we call that as a dominance relationship. So simple dominance relationship, let's say, between alleles at a locus, right? So, and you might have learned this in a very different way, right? You might have learned it as, here's a red color and a white color, and the red color is dominant compared to the white color, right? That's very simplistically putting that, right? I want us all to sort of move away from sort of thinking about it in terms of phenotypes and think about it in terms of the genome itself, right? So in this case, if we think about, if we just analogously compare this at the genomic level, we're saying, let's say an A allele, let's say, happens to control the red color variant, and let's say a C allele happens to control the white color variant. And if you seem to observe greater degree of that red variant in that population, maybe we say that the A allele happens to be dominant over the C allele. Does that make sense? Right. So in this case, we're talking about allelic relationships or allelic dominance relationships. And there's different types of dominance relationships. Right? We have what's called complete dominance, right, where one allele is almost entirely dominant over the other allele. You have over-dominance. You have incomplete dominance, where you show some sort of like in between variant, right, when you have a heterozygote at that locus. And then there's there's different kinds of dominance, pleiotropy, epistasis, et cetera, et cetera. We'll not go there. Okay. The second principle is called the principle of independent segregation. And the second principle refers to the fact that alleles at the same locus, in other words, homologous alleles at the same locus, segregate away from each other during mitosis. And this specifically happens, if you think about it, during anaphase, either of the anaphases, right? So anaphase 1 or anaphase 2 of meiosis. When homologous alleles essentially move away from each other and are distributed or segregated into independent cells. So this is basically saying mom's copy and dad's copy essentially segregate away from each other. Again, this is all, we're still dealing with sexually reproducing diploid species. Okay. Homologous copies segregate away from each other. Okay, so that's the second principle. And then the third principle is what's called independent assortment. And so this is when you think about multiple loci and alleles at multiple loci at a time. Mendelian principles state that loci are independent of each other, which means that a locus that controls, let's say, hair color segregates or assorts independently of a locus that determines skin color or so on and so forth. I'm just simplifying this for the skin color and hair color are polygenic, multi-allelic traits. Okay, but that's why I said these are Mendelian traits or Mendelian loci, which means that they are controlled by usually monogenic biallelic loci. So independent assortment refers to alleles at multiple independent loci assort separately, right? We call this as independent assortment. Okay, and this is important because when we discuss sort of basic population genetics, this will become very sort of important. Okay, now let's jump a little ahead and let's define a population. What's a population? Yeah, group of individuals. Yeah, same space, same time, usually of the same species. Biological species concept also tells us that a population has to mean that they interbreed with each other, right? They are essentially mating with each other, passing their genes on within that spatial and temporal sort of scheme, right? So population is just individuals of the same species. So individuals of the same species that interbreed with each other, right? In space and time. Let's just call it sort of generally speaking, right? In space and time. We're just kind of interbreeding with each other. Okay. All right, so let's go ahead and use these ideas that we've discussed so far. I'm going to do some mathematical representation of all of these concepts. Okay. And this will become relevant because this is sort of what I call a null hypothesis for everything in evolution. And you might have we actually discussed this earlier on when we were talking about determining genotype likelihoods. And this is the idea of what's called Hardy-Weinberg equilibrium, or the Hardy-Weinberg principle. It's a very straightforward sort of principle. So let's consider a single population. So population of individuals. So one population, let's say at time t, and this is not relevant for this purpose for right now, but time t will become relevant when we think about the next ideas. Let's say at time t in this population, let's say that you happen to have a single locus at a single locus. And let's make it even simpler. Let's make it a biallelic locus. So which means there's only two alleles at this locus, A and a C, or A and a G, or something like that. Let's say the two alleles happen to be A and G. Okay. Again, let's also assume diploid, sexually reproducing species here. Okay. Just for ease of doing the math here. Let's assume at this time t, I'm going to say the, let's make this easier. The total number, you go ahead and you genotype these individuals. In other words, you sequence a bunch of them, you call variants, and you've determined all the genotypes at that locus. Okay. And say the total number of AA, right? These are called homozygote AAs, right? So you have the total number of homozygote AAs is something, right? So let's call this as just NAA. And let's say the total number of AGs, let's call this as NAG. And the total number of GG individuals, let's just call this as NGG, right? So this is the number, physical number of genotypes that you've determined after you've called variants, right, at that locus. This means, right, let's say that N is just the total number of individuals that you've genotyped in this population. So it's just N each one of these, right? So you add them all up, and that gives you N, right? Now this means that the frequency of AA individuals in this population is just this, right? NAA by N. Frequency of the AG individuals is just NAG by N. And the frequency of GG individuals is just NGG divided by N, right? So we call these as genotype frequencies. In that population, right? So these are the frequencies of that genotype in that population. So one way of summarizing your data, instead of just looking at the numbers, right, the raw numbers, you can compute what are called the genotype frequencies. And this gives you a summary, if you might, right, of the relative proportion of one genotype compared to everybody else. Now you tell me, now that we've discussed a few of these concepts, what is this genotype frequency indicative of? Think back to Mendelian concepts or Mendelian principles. What if I have AA genotype frequency as 0.9 and the AG as 0.1 and GG as 0, right, in that population? What is that potentially indicative of? Right, and can you think of why would 90% of that population have AA? It's dominant. Maybe it's dominant, right? Maybe it's dominant. Or maybe it is being selected for, right? Maybe AA homozygote individuals happen to have greater degree of fitness compared to others. Or the presence of the A allele seems to confer some sort of different fitness differential for those individuals. Or maybe it's the other way around. GG individuals are at a disadvantage, right? They just die off faster, and so they don't actually survive to be able to pass their genes on to the next generation. Or maybe there's inbreeding within that population, and AA individuals only breed with other individuals that carry an A allele, right? So there's a lot of host of different possibilities that could occur, okay? But just thinking about it, right? If you observe greater degree of genotype frequency of one genotype compared to others, right? Simplistically put, that could indicate some sort of a dominance relationship, right? This is what Mendel observed, right? When he was doing his experiments with bean plants, and he tended to observe only a certain greater frequency of a certain one particular phenotype in that population after generations and generations of it. That's how he decided that, hey, guess what? Maybe this one particular red flower or round pea shape seems to be much more dominant compared to others. So genotype frequencies are usually indicative of dominance relationships. Simplistic, if not assuming nothing else is happening. So there's another way of summarizing data, and this is probably the most common way of summarizing data, and that is using what are called allele frequencies. And so very easily, you can compute the allele frequencies from your genotype frequencies, right? So in this case, if you're calculating the frequency of the A allele, let's just call it P of A. Again, the reason I'm using the probability notation here is because frequency and probability are all referring to the same thing in this case, right? Because frequency of observing an AA individual is nothing but the probability that you close your eyes and you randomly pick an individual and that individual happens to be AA, right? They're the same thing. That's why I'm using the probability notation here, okay? So P of A, which is the probability of randomly observing an A allele, is just, in this case, right, if you think about it, since every AA individual happens to have two A alleles, right? So this would be 2 times NAA, or you could have an AG individual who would have only one A allele. So it's 1 times NAG divided by, since these are diploid individuals, 2n, right? So this is the total number of alleles in this population given n diploid individuals. Right? Now you can simplify this a little further, right? So this would be just nothing but probability of AA plus 1/2 probability of AG. So you can simplify this a little further. So in other words, if you have the genotype frequencies, you can easily estimate the allele frequencies. It's not the other way around, unfortunately. And we'll talk about how that is so in a second. So similarly, if you do the math, this is just equal to 2 times NGG plus 1 time NAG divided by 2n. So this is just probability of GG or genotype frequency of GG times 1/2 times AG. Probably. Okay. By the way, I forgot to mention, right? So very importantly, since these are again probabilities and they are mutually exclusive events, right? If an individual is AA, they can't be AG at the same time or GG at the same time. So which means these probabilities, right? So P of AA plus P of AG plus P of GG equal to 1, right? So this is a truism. Sum of all genotype frequencies must always add to 1. Similarly, P of A plus P of G must always be equal to 1. Again, these are mutually exclusive probabilities. Okay. So we've already discussed 2 summary statistics, right? We call these as summary statistics because think about it, right? Let's say you have 20 billion genomes, right? How the heck are you going to sort of, whittle down all of that information into understanding what is happening at the population level? Okay. So clearly you need some sort of a information sort of, sort of dimensionality reduction way of kind of understanding or summarizing these data. And so that's why you use summary statistics, right? So these are 2 very commonly utilized summary statistics or data representations of your populations. Okay. Now remember, when you're thinking about, say, things like SARS-CoV-2, for instance, there is no genotype per se, right? Because these are haplotypes, right? They're haploid genomes. So you wouldn't have genotype frequencies, you would have allele frequencies instead, that's all. Okay. So two common ways of summarizing data sets. Got genotype frequencies and allele frequencies. Cool. Now, just quickly, let's go ahead and derive sort of what exactly the expected probabilities of genotype frequencies given your allele frequencies are. And this is that principle that we talked about, the Hardy-Weinberg principle. So if I assume that, let's say this is P, just for ease of a sort of nomenclature and just kind of, making you all remember some of these concepts a little easier. Let's say the frequency of the A allele, let's call it P. Frequency of the G allele, let's call it Q. So P plus Q is always equal to 1, right? In this case. And it's assumed that this population is randomly mating, and that's very important. So you undergo one generation of random mating. So what that means is, in this case, an individual that carries an A allele has equal propensity or probability of mating with an individual that carries a G allele. So there is no fitness differential. A's aren't better, G's aren't, worse, etc. They're all the same, equal probability of mating. Everybody gets to mate with everybody else. So if that occurs, what's happening, right? If we think about sort of gametic mating, right? The gametes can either carry an A or a G. That's it, right? You either have A or you have a G. And why is that true? That is because of the independent segregation principle, right? So this principle right here tells me that homologous alleles segregate away from each other. So if an individual happens to be AA, the A and the A allele, mom's copy, dad's copy, will segregate away from each other. If the individual happens to be AG, then the A and the G will separate away from each other, and so on and so forth. So if that holds true, then what can you do when you say that this population is randomly mating? We can draw that Punnett square, right? That Punnett square is essentially allowing you or permitting you to represent random mating, assuming that this mating process of an A with an A or an A with a G or a G with a G are independent events. Right? So in other words, let's say the maternal copies happen to be either A or they happen to be either G. Similarly, the paternal copies in this case happen to be either A or they happen to be G. And the frequency with which you would observe this, right? P of A is just P. P of G is thus Q. Similarly, P of A here is P, and P of G here is Q. Right? So in other words, if an A gamete mates with another A gamete, you produce an AA homozygote offspring. And the frequency or the expected frequency with which you would expect that to occur if everything is random, independent segregation, independent, dominance relationship between P and Q, it would be P times P, which is P squared, right? So because this is just equal, the probability of obtaining in the next generation, T plus 1th generation, an AA homozygote would just be equal to probability of A in the previous generation multiplied by probability of A, which is equivalent to P squared. Note that here I'm using the T to represent time, right? So here I'm saying at time T, the frequencies are P and Q. At time T plus 1 in the next generation, what are the expected genotype frequencies? So similarly, PT plus 1 of GG, right? This guy right here, the expected frequency would just be equal to Q squared, right? So this is just P of T of G, PT of G, which is equivalent to Q squared. Now, AG, on the other hand, sorry, excuse me. Yeah, okay. Yeah. AG, on the other hand, right, can occur in two possible ways, right? You could either have maternal A join with paternal G, or you could have the other way, right? So you could have AG here, or you could have AG here. And the probability with which each one of these could occur is PQ, right? So then the probability of T plus 1 AG, which is the heterozygote frequency, is just PQ plus PQ, right? And why do we do plus? That's because these are mutually exclusive, right? They can't happen at the same time. They're kind of like addition rule of probability. And this would be expected to be equal to 2PQ. And so we call this as sort of the first principle of Hardy-Weinberg equilibrium, which is basically telling you that the expected genotype frequencies in a population that is randomly mating, assuming all of the other stuff, right? So assuming frequencies are present at P and Q, independent segregation of alleles would be expected to be P squared, Q squared, and 2PQ. Okay. That's the sort of first principle of Hardy-Weinberg equilibrium. Now, the second principle of Hardy-Weinberg equilibrium is actually much more important in my head, at least. And that is what happens to the allele frequencies after one generation of random mating. And this is very easily predictable because remember, we just derived this. You can calculate the allele frequencies easily from the genotype frequencies. Right? This is the formula for doing that. So let's go ahead and plug those in to obtain the allele frequencies. So PT plus 1 of A would just be equal to PT plus 1 of AA plus 1/2 of PT plus 1 of AG, right? That's just the same formula that I had up here. I'm just plugging this in into my equation. And so if I go ahead and do that, this is just equal to, in this case, P squared plus 1/2 times 2PQ, right? So this is just P squared plus PQ, which is just P times P plus Q. But what do we know about P plus Q? P plus Q is equal to 1, right? Allele frequencies must always be adding to 1. So in other words, the allele frequency in the next generation stays the same. So the frequency of the allele after one generation of random mating always stays the same. Okay. So similarly, PT plus 1 of G would be expected to be equal to Q, which is just PT of G. And we call this as the second principle of Hardy-Weinberg equilibrium, which basically tells you that after one generation of random mating, the allele frequencies do not change. Okay. So far, so good. Okay. And so I like to call this sort of, like I said, the null hypothesis for a population that is not evolving. And this is very important because if a population has to evolve, allele frequencies have to change. Okay. If allele frequencies are not changing over time, they're just constant over time, we say that that population is not evolving at that locus. Okay. So in other words, this is sort of leads to a definition of what we, like to call evolution, but a much more nicer population genetic way of defining evolution. Evolution is frequency or allele frequency change over time. So in other words, in order for evolution to occur, the population should not be in Hardy-Weinberg equilibrium. Okay, it's the other way. Cool. So far, so good. Okay, so let's do a little sort of, these are sort of tests of Hardy-Weinberg equilibrium and stuff like that, which we'll discuss when we do lab, honestly. The idea, there's folks might have learned a couple of tests at least. There's a simple test called the chi-square test of Hardy-Weinberg equilibrium, which is probably the most commonly utilized one, where you basically take the observed values, right? So in this case, an observed value of all the genotype frequencies in a population. And then you compute the genotype frequencies, and then you compute the allele frequencies from that. If you assume that that population is randomly mating, in the next generation, you can easily calculate the expected genotype frequencies, which are P squared, 2PQ, and Q squared. So you've got your observed values, you've got your expected values. You can compute a chi-squared statistic, which is non-normalized chi-squared statistics is just observed minus expected, this whole square divided by expected, and you sum all of that. And then you calculate the total number of degrees of freedom. In this case, since you have only 2 alleles, there's only 1 degree of freedom. Okay. So then you can go back to your chi-square table like I showed you earlier today, and you calculate your critical value from that and determine if that population is in Hardy-Weinberg equilibrium or not. Again, my goal here is not for you guys to do that on paper. We'll actually learn how to do that using R or some summary statistic packages and whatnot using whole genomic data. Because you can imagine if somebody were to sit there and compute that across 30,000 loci, right, you're gonna, you're gonna get old. You'll never get it done. Okay, so that's why this is a computational genomics class. So we'll learn how to do that, sort of all these massive tests of Hardy-Weinberg equilibrium within seconds. Okay. So that's the first concept that I wanted to go over. Now let's talk about sort of some other sort of assumptions that we make when we think about a population that's in Hardy-Weinberg equilibrium. So when we assume that a population is in Hardy-Weinberg equilibrium, the first one we made was random mating, right? So in other words, individuals within the population don't tend to sort of have any fitness differential, or one individual doesn't get to preferentially mate with another individual, right? We're assuming everybody gets to mate with everybody else. So if a population is in Hardy-Weinberg equilibrium, we assume random mating. There are some other assumptions that we commonly we make. We assume that there is no mutation. So in other words, there are no sort of new mutations showing up into this population, right? So we assume that if an locus is biallelic, only 2 alleles, it stays that way. There's no third allele that just randomly pops up. We know that's not true, right? Mutations happen. Another assumption we're going to make is no migration. So let's assume that this is still a single population and there is no individuals leaving this population. There's no individuals coming into this population to change the frequency of those alleles. Okay? So population size, in other words, is held constant. Another assumption that we're going to make is a large population. So we're going to assume that these populations are sort of infinitely large. So we're going to go ahead and say that, since these individuals are, these populations are so large, right, even minuscule changes in allele frequencies are not really showing up over time. So these are large populations. Another assumption we're going to make is no selection. So in other words, one allele is not better than any other allele, or one genotype is not better than any other genotype. All alleles, all genotypes have the same fitness. So all of population genetics really, right, at least theoretically thinking about it, talks about deviations from Hardy-Weinberg equilibrium. So each one of these cases, and we know none of this is true in natural populations, right? No population is just entirely randomly mated. No population is infinitely large. Every population has selection. Every population has migration. Every population has new mutations accumulating over time. So we know none of this is true, right, in real populations. So what we're going to do is we're going to spend a little bit of time thinking about sort of theoretical deviations from each one of these assumptions. Okay. So far, so good. So let's start with the first one here. Let's start with large populations, and that's sort of the easier. Well, I don't want to say easier, but it's very intuitive to understand large populations versus small populations. So let's consider a simple case where you have a. So let's take this. Let's mark this. So first one is small populations. Again, small is in quotes here. Let's just call it as a finite population. Okay, so in other words, it's not infinite. It's a finite population, and let's say that the total size of this population is n. Okay, so you have n total individuals who are diploid biallelic at the same single locus. And let's say that this is the population at time t, and let's say at time t plus 1, it is this generation, this population. And let's assume that we're going to say sample individuals or sample alleles with replacement until we hit n total individuals in this t plus 1 generation. In other words, we're just going to repeat this process of sampling from one generation to the next. Okay. So here, let's write down some stats here. Let's say PT of A allele is P. And say PT of the G allele happens to be Q. And in this case, this is just equal to NAA. Sorry. This is just equal to 2NAA plus NAG divided by 2N, right? So this is the same formula that we wrote earlier. And let's say that from this generation to the next, right? So all we're doing is we are sampling with replacement into this next population. And let's say during this process, right? Let's say I have sampled a total of X A alleles and 2N minus X G alleles. Right. So X is the total number of A alleles that I've sampled, and 2N minus X is the total number of G alleles that I've sampled, right? Why is it 2N minus X? It's because this is the diploid population size. Then you have 2N total alleles to sample. So then in this generation, PT plus 1 of A would just be equal to X divided by 2N. And PT plus 1 of G would just be 2N minus X by 2N, right? So this is the frequency of these alleles in the next generation. Okay. By the way, this process, right, very simple way of sort of quantifying evolution. We call this as a Wright-Fisher process, named after Sewell Wright and Ronald Fisher, who both sort of discussed the theory behind genetic drift, which is kind of what this principle that we're talking about is. Okay, so if this occurs, as it turns out, right, this is very equivalent, right? This is just a Bernoulli trial, right? Remember, we've talked about Bernoulli trials in like 2 or 3 different contexts at this point, right? So Bernoulli trial is because you either have one or the other, right? In this case, you either have an A allele or a G allele that you're sampling from one generation to the next. The probability of that occurring can be calculated using the binomial probability. And so the binomial probability is just written here is the probability, let's say in generation T plus 1, I have X A alleles and 2N minus X G alleles, right? Given, right, P and Q, right, in the previous generation, can just be written as the binomial probability, which is just 2N choose X, in this case, P to the X, and Q to the 2N minus X, right? So this is just the binomial probability from one generation to the next. Can be just written using this formula, right? So 2N choose X, P, which is the frequency of the allele in the previous generation, to the X, right? So you have X total copies of the A allele, Q to the 2N minus X. And by the way, Q is 1 minus P, right? So really there's only 1 degree of freedom here, right? Because there's only one parameter. If you have P, you can get Q. So this is just 2N choose X, P to the X, 1 minus P to the 2N minus X. We call this process, right, of just sort of random fluctuation in allele frequencies from one generation to the next, because just by chance, you could pick more of the A allele. Just by chance, you could pick less of the G allele. We call this process as genetic drift. Okay, so genetic drift is sort of what we call stochastic or random fluctuation in allele frequencies from one generation to the next. So there's nothing causing this A to be picked over G. Okay, it's just random. Close your eyes, right? And just randomly pick alleles. We call this process as genetic drift. And so you tell me, right, looking at this simple equation, what are the things that determine the rate of genetic drift? What are the factors in this equation right here? X is independent, right? X is random, which means 2n minus X is also random, right? So what are really the things that are determining the rate at which drift is happening?
Population size.
Yep, population size, which is this guy, right? So you got N, which is the population size. What's the other factor? P, right? Which is the frequency of observing the allele in the previous generation. So really, if you think about it, the rate at which drift happens is determined by two things. So rate of drift right, is determined by one, the population size. Here that is N, right? And it is also dependent on P, which is the frequency of the allele in the previous generation. Okay. And let's understand this. Let's think about this process. Let's do an example, for instance, right? Let's say that N in this case, let's say happens to be equal to 100. Okay. So let's do an example here. Let's say N is 100. And let's say here P, which is the frequency of the A allele in the previous generation, let's say it is 0.5, which means, this is P, which means Q is also 0.5, right? So that's the frequency of the other allele in the generation. So this means, let's say that the probability, I want to know the probability that I just randomly lose this A allele in the next generation. Okay. So I want to calculate the probability that X equal to 0, right, in the next generation in T plus 1, right? You can calculate this using the binomial probability. So this would just be equal to 200. Sorry, let me use, for convenience's sake, let me use the same notation. Right? So little n here. So 200 choose 0, right? Because I have 0 copies of the A allele in this case. 0.5 to the 0 times 0.5 to 200, right? So this is just the probability in this case that I randomly lose the A allele after one generation of drift, right? Just by chance. You can calculate this, right? Very easily. Let me do this in R quick. So 200 choose 0 is just 1, by the way, right? So anything choose 0 is 1. So all I need to do is just 0.5 raised to the 200, which is a pretty small probability, right? So it's this happens to be 6.22 10 to the minus 61. Okay, pretty small probability. So I'm not going to randomly lose this A allele just by chance, or probability of that happening is very rare, right? In this case. Now, on the other hand, if n equal to 10, in the same example, if I have n equal to 10, right, then what happens? In this case, and let's assume everything else is the same. So let's say P here is 0.5 and Q equal to 0.5. Then this probability of X equal to 0 in T plus 1 would just be equal to, in this case, 0.5 to the 20, right? Which happens to be, right, 9.54 10 to the minus 7. Now just look at these two numbers here, right? Compare this guy versus that guy, right? And all I'm trying to do is just to make a point about population size and the rate of drift. So which one is greater, or which one is more likely to occur? Yeah, population of 10, right? Because this, of course, this number is also very small, right? But if you compare it to 10 to the minus 61, right, 10 to the minus 7 is probably much more likely. Right? So in other words, populations that are much smaller are more likely to be affected by drift. So the point I wanted to make here is that smaller population size implies greater degree of drift. And so greater chance that randomly some of these alleles would be lost. Okay. Cool. Perfect timing because it's 9:15. So I'm going to stop here. Okay. And what we're going to, Will, you had a question. Go ahead.
I guess we could wait till after class. That's
Okay.
fine.
Sounds good. Great. So what we're going to do is when we come back on Friday, we're going to discuss a few more of these deviations from Hardy-Weinberg equilibrium. So we'll discuss about, sort of specialistic cases of mutations, migration, selection. How do you kind of quantify all of those things? And importantly, we'll learn how to summarize all these data sets, right? And there's some very nice ways of summarizing these data sets, including what's called the allele frequency spectrum. We'll learn a little bit about that and why that's relevant, particularly in this case, let's say to study what's happening, the evolutionary trajectory of, let's say, SARS-CoV-2. How do we determine that in this case? Okay. And that's very important, right? Because if you are a CDC epidemiologist, you are curious to learn what is the evolutionary trajectory, right? How is this virus evolving? Is it just randomly drifting, or is it actually evolving in response to, in this case, vaccination status, or are there any particular new novel variants that seem to be much more fit compared to others, right? So, if you were Pfizer or Moderna or the CDC, that's what's important to you, right? Because you want to be at the frontier of trying to determine what is the next strain that becomes much more virulent, right? So that's why this is relevant. Okay, so we'll learn about sort of how this information whittles down to the idea of SARS-CoV-2 later on as well. Okay, sweet. Thanks everybody. We'll see you all on Friday. And like I said, if you're want to just finish up the likelihood ratio test and just upload it, just go ahead and do it. That's fine. Yep.
I mean, I guess I'll ask.
Thank you.
Will.
Yep.